Let's screw this pathway up now. Let's see how this thing goes in terms of cancer biology.
So this pathway stimulates cell division and development?
Yeah. And tissue repair and stuff like that. For instance, if you cut yourself or whatever, you can thank, in part, elements of this pathway for stimulating that cell division and repairing, mitosis and repairing the damaged tissue. Okay, now, how can we screw this up? It turns out that virtually everything on this pathway that we have seen, if things go wrong, can push a cell to a cancer state. In other words, the genes for these are all called onco, or cancer genes, when they're mutated stuff. Now, fortunately for us, there's only a few we could call mutational hotspots that are commonly found in a lot of cancers. And we'll see that in one moment. Oh, by the way, I forgot. What do these things stand for? MAPK means, let me put this down, mitogen associated protein kinase. And MAPKK's protein kinases. In other words, they phosphorylate the MAPK. Now, what is a mitogen? A mitogen is something that generates mitosis or stimulates cell division. So things that stimulate cell division are called mitogens. And when they started getting into molecular biology and biochemistry, they would find the activation of these protein kinases when you stimulated cell division with some kind of chemical agent. So that's what the term stands for. Okay, well, let's go back to the issue of cancer biology. Couple major hotspots. The first one is right over here, old G protein RAS. RAS mutations are involved in about 30% of human cancers. That's a lot of them. And it turns out what happens with RAS,
What percentage?
about 30%. What happens with RAS is, for some reason, you frequently get a deletion of part of the gene at one end of it. Unfortunately, deleted part contains the active site for the GTP hydrolysis. So these RAS mutations can no longer hydrolyze GTP. And of course, hydrolyzing GTP is essential for active RAS to go back to inactive RAS. So what that means, if you take one of these guys and you turn it on, it's got no off switch. It continues to activate this pathway long after the growth factors are gone. That's not good, because the growth factors are secreted by cells telling, okay guys, it's time to divide, or it's time to differentiate. So many times RAS mutations, what's going to happen there is that they stimulate cell division when you don't want it to. Unfortunately, we don't have any drugs that inhibit RAS yet. I'm sure people are working on that, and they know the tertiary structure of RAS in both forms in a great amount of molecular detail, but they haven't found anything that can shut this thing off when it's overactive. Okay, so that's probably the worst hot spot here. But another one of the families of growth factor receptors, specifically the EGF, or epidermal growth factor receptor, which is involved in a lot of mesodermal type tissue development, things like that. There are common mutations that allow the EGF receptors to bind together and phosphorylate each other even when there's no growth factor around. That is a really bad thing, because of course you can activate this whole pathway even when there's no growth factor present. Bad idea. Okay, that's really bad. Now, There actually is a specific biotechnology drug that works by binding to these mutant EGF receptors and basically preventing them from binding to each other and phosphorylating each other. And that drug, which is used in numerous different kinds of cancers, is called Herceptin. That's the commercial name. It's been around for a number of years now. It's used in a number of different kinds of cancers. There's a genetic test to see whether you have that particular mutation in the EGF receptors. And if they do, that's part of the standard kind of treatment, in addition to chemo and all kinds of other nasty stuff. But this is used. It's not a cure by any means, but it does slow things down in many cases. Okay, so that's another hotspot. Now the third hotspot is right over here. The Raf kinase. Now Raf stands for RAS-associated factor. Once again, a factor is something that does something, but we have no idea what it is yet, at least when it was first discovered. All they knew was some kind of protein bound to RAS, bound to active RAS, and they had no idea what it was until later. Turns out, of course, it's a family of kinases. It turns out one of the RAF kinases, called RAF-B, is involved in 2/3 or more of human melanomas. Okay. Now, it turns out in many of these melanomas, there is a mutation in that RAF-B kinase that keeps it switched on all the time, even when there's no active G protein RAS present. Needless to say, that is not good. Now, the problem with melanomas are this. Melanomas, of course, are cancers of the pigment containing skin cells called melanocytes. And that's what gives people dark skin or light skin or what have you. People that are very dark skinned, like folks from African or ancestral Australian ancestry, have lots of melanocytes in their skin. Us white boys and girls from Europe, especially Northern Europe, have very few because there's no sun out there. When it is, it's way down the horizon anyway. And if you come from Britain or Ireland, it's always shrouded in clouds. So, okay. So, the problem with these things, the problem with melanocytes is when they go bad really fast. That's why I tell you, if you have any crusty moles or irregularly shaped moles or you're bleeding from a mole, go straight to a doctor because it proliferates, these things proliferate fast. They also have a nasty tendency of very quickly getting on and invading other tissues, going in your bloodstream and setting up satellite colonies all over the place. And when you get to that point, when it's metastasized, you're basically toast. Okay, now the thing is, if it's early before it's spread, they just dig out the thing and then you're hopefully okay. If it has spread, you're in serious trouble. Now the problem with melanomas is these cells, the melanocytes in the skin, and also the glial cells that are support cells in the central nervous system in the brain, and then other cell types, those cells are derived from a group of cells called the neural crest cells. Now what happens is early in development, when the neural tube is forming, some cells at the very top of the neural tube, when they intersect, when the neural groove folds over and intersects with the other side, some of the cells at the top, which we call the neural crest, the top part of the neural tube, start migrating all over the embryo and proliferating in different locations, turning into parts of your jawbone, some endocrine glands, melanocytes in the skin, the glial cells in the central nervous system, and those kind of things. But the problem is, those types of cells tend, in a sense, because they can form both neural type cells, they also form your peripheral nervous system. The neural crest cells have the capability of differentiating into many different cell types, all the way from nerve cells to sort of cells that are halfway in between, like some of the, what they call them, neurosecretory cells and some endocrine glands that have characteristics of both, to flat-out mesodermal type cells. In other words, they're almost like a form of stem cell. So you can imagine when these guys go bad, they kind of revert back to their childhood. What are they supposed to do in embryonic development? Go all over the embryo and start proliferating. That's really bad when you're an adult organism. They go back to childhood. Oh, we're supposed to spread all over the place and proliferate. and then it kills you, okay? All right, well, RAF-B, melanomas, most melanomas. Just last year, 2 FDA-approved drugs are used that specifically inhibit RAF-B are on the market. It's been some years in clinical trial. About 5 years ago, they found that the RAF kinases were heavily involved in melanomas. The drug companies already had the tertiary structure of the protein, started going and experimenting, trying to make some drugs that could block it and not block other kinases. So they came up with some, went through FDA things. 2 are on the market now. There are more in the clinical trial pipeline. So that's actually a very useful kind of thing. Now, it does not cure it, but what it does is slow it down. Generally, they just use it. It's extremely expensive. Drug companies make as much profit as they can. Generally, they just use an extreme, in late stage advanced melanomas. And generally, people only have a couple months left with that. This will stretch out to 6 or 8 months. It's not a cure, but it does stretch things out, and it has relatively few side effects, unlike the other stuff they use on that. So, lots of investigations on this pathway. Here you have any of these things in this pathway, when they go wrong, are potential cancer causers, but most of the mutations and the other stuff are fairly uncommon. Yes?
So when you have like, skin pigment problems, that's from that? I mean, that's all from that? Or?
Well, not necessarily. It's a bunch of other things. If you have the thing where part of your skin develops pigment and part of it bleaches, I don't know what causes it. I forget what causes it, but I don't think it has anything to do with it.
I had that on the bottom of my leg.
Oh, really?
Foot, and I don't know what causes it.
Yeah, that kind of thing is kind of common. That or sometimes sort of red areas. Remember former Russian President Gorbachev? He had that big red mark there. Yeah. I guess a lot of the former old communists thought that was the mark of Cain or something. They didn't like it. Their whole country collapsing. Okay, but at any rate, so this is clearly a very important, developmentally important type of signaling pathway. And we're going to see, aside from this pathway, there are other ways of activating. That's going to be very important. Another type of thing that's a signaling agent as well as playing other critical cellular roles. We'll see that later on. Okay, well, we're going to look at another signaling pathway, and then we're going to go on to something that has important signaling roles. Okay, any questions about this? And here again, I know we went through this for folks that just took or remembered cell recently.